So good afternoon, everyone. And I've been asked to talk about oncology and inflammation today. And so, so the learning objectives are for today are to understand the role of inflammation in the cancer phenotype and to show how imaging inflammation and the phenotypic changes induced by inflammatory pathways can be applied to understand cancer biology and the role of inflammation in cancer. And then finally, I'd like to talk a little bit about image-guided targeting of inflammatory pathways in cancer. So the word inflammation means I ignite or set alight, and it is part of the complex biological response of tissues to harmful stimuli, such as pathogens, damaged cells, or irritants. And inflammation is a protective attempt by the organism to remove these stimuli and to initiate the healing process. So it occurs all the time in tissues. But cancers, of course, as you've heard earlier, they are this highly heterogeneous, complex organizations, and they typically have these wound-like environments. So they have hypoxia, they have acidic extracellular pH, and in 1986, Harold Dvorak actually came up with this hypothesis that tumors are wounds that do not heal. And these hostile or wound-like environments invoke this inflammatory type condition in many tumors. So a little bit about the inflammatory response. So the key molecules that affect this inflammatory response are called prostanoids. And prostanoids are formed by the breakdown of arachidonic acid in membrane phospholipids by the hydrolyzing action of phospholipases. And so arachidonic acid is metabolized by lipoxygenases, P450 enzymes, and by cyclooxygenases to create these prostanoids that play a host of roles in the mediation of inflammation. And of course, this enzyme, or actually there are cyclooxygenase 1 and 2, are the ones that are the key mediators of the inflammatory response. And you get these several types of prostaglandins and thromboxanes, and each of these have receptors that mediate these biological effects. And so these receptors are expressed on these different tissues, and then the prostaglandins bind to these receptors, and you get these very specific responses. Of course, in tumors, when you have these prostanoids being formed, it also mediates a crosstalk between the tumor cells, the stromal cells, and the endothelial cells. And again, the prostanoids, primarily through the cyclooxygenase reaction and the formation of PGE2, then result in modifying angiogenesis or resulting in increased migration. You get changes in cell proliferation, and this occurs through both autocrine as well as paracrine signaling in cancer cells, and with crosstalk between stromal cells and endothelial cells and cancer cells. So just to summarize, so the response of living vascularized tissue to injury is inflammation. And for inflammation, arachidonic acid is converted to various eicosanoids by the action of lipoxygenases and cyclooxygenases. And most solid tumors exhibit inflammatory properties that are characterized by levels of prostaglandins and pro-inflammatory molecules. Now, the enzyme in cancer that seems to play a dominant role is cyclooxygenase 2. So the cyclooxygenases, so there's 1 and there's 2. Cyclooxygenase 1 is the constitutive form, 2 is the inducible form. And it's 2 that plays the major inflammatory role. And PGE2 is the major product that mediates inflammation in tissues. So now, of course, as you've all heard before, there are various anti-inflammatory agents. So these are non-steroidal anti-inflammatory agents that can bind to cyclooxygenase 1 and 2. And so these are, your typical aspirin, indomethacin, and now there are also selective COX-2 agents. So COX-1 is not affected by the inflammatory process, and in fact is gastroprotective. COX-2, on the other hand, is not detectable in normal tissue, but is detectable after induction by inflammatory stimuli. And so if you do a quick literature search, so if you see inflammation and cancer, you have about 20,000 hits. And then if you look at COX-2 in cancer, there's about 5,000 hits, even more than HIF in cancer. And of course, there's a great deal of interest in the role of cyclooxygenase 2 in cancer. And there are several clinical trials that are ongoing and that have occurred in the past. They have mixed outcomes, but part of it is that these cyclooxygenase COX-2 inhibitors, of course, create also side effects. And that is partly because they can inhibit COX-1 as well, and that creates gastrointestinal problems. And of course, the other thing is that they're not very specific and have other side effects as well. Just not only inhibiting COX-2, but then affecting apoptosis and other pathways. So now, as you can imagine, there's a lot of interest in imaging cyclooxygenase 2 in cancer. And the group that has led the field is at Vanderbilt University, and they have been developing imaging agents to detect COX-2. So if you can visualize tumors that have high COX-2 expression, then you can choose them for COX-2 inhibition. And so the group at Vanderbilt have used COX-2 specific inhibitors and have then put fluorescent moieties on them and created ways to visualize COX-2 expression levels. So here they've taken Coxib, which is a COX-2 inhibitor, and then called it fluorocoxib. Just 2 examples of this. And then, of course, if you can visualize COX-2 with molecular imaging, then you can start to target it and see its downregulation non-invasively in vivo. And so here are examples from their work. So here is an image of a mouse where inflammation was induced in the paw using this inflammatory agent, carrageenan. And you can see that this mouse, when injected with fluorocoxib, you see this retention of this imaging agent in the inflamed paw, as shown here. They've also tested this in tumor models. And so here is a mouse with a tumor that expresses high COX-2, and here's a mouse with a cell line with no COX-2 expression. And here you can see the retention of this COX-2 agent, inhibiting agent, in the tumor model with high COX-2 expression, showing that it's possible to visualize COX-2 expression levels non-invasively. And this work was presented by them in Cancer Research a couple of years ago. And these COX-2 imaging agents are now commercially available. Of course, you can use imaging to visualize COX-2 expression, but you can also use multimodal imaging to look at the effects of COX-2 downregulation on many of the phenotypic characteristics of tumors. And so to do this, you can create cell lines that express short hairpin RNA against cyclooxygenase 2 that completely wipes out COX-2 in cancer cells, as shown here, where these triple-negative MDA, MD231 breast cancer cells express short hairpin RNA. And here you can see that MDA231 cells, when they're exposed to this inflammatory agent interleukin or TPA, show this robust COX-2 expression. Sorry, this pointer isn't working. But if you take cells that express short hairpin RNA against COX-2 and then expose them to TPA or interleukin, you see no expression of COX-2 in these cells. So then you can start to use imaging to understand the phenotypic effects of this target. And so here, if you take these cells and you put them in vivo, they don't grow. And throughout the earlier two talks, we've observed the heterogeneity and the complexity of tumors. But of course, if you can find that Achilles heel in tumors and find a target that is so important to cancer cells that if you knock it out, the cells stop growing or stop metastasizing, of course, that is one of the key issues in cancer, because there are so many ways that cancer cells adapt and survive. So it's a question of the Gordian knot versus the Achilles heel. And maybe for some cancers, COX-2 is a very important target. And so here, these tumor cells did not grow in vivo, as shown here. If you do a molecular characterization, what you see is a host of oncogenes get downregulated, and a host of suppressor genes get upregulated. So now this is a single enzyme that has this profound target on so many pathways. And importantly, if you take these cancer cells and put them into the tail vein, what we find is that they do not metastasize, as shown here, where these are the wild-type MDA231 cells that show a lot of metastasis in the lungs. But if you knock down COX-2, you hardly see, in fact, there was no metastases in these lungs of mice injected with these cells. Of course, you can use imaging, and if you use something like MRI and MRS, you can use it to look at cells, you can use it to look at preclinical tumor models, and of course, you can use it clinically, again, as highlighted in the earlier two lectures. So here, what is possible to do is actually look at the effects of these cells in a cell perfusion system that has been adapted to be able to detect the ability of cancer cells to degrade matrigel, which is extracellular matrix, which is typically what cancer cells degrade when they metastasize to distant sites. And here, what was observed is that if you take these cells and you layer them on matrigel and you visualize the degradation of matrigel non-invasively with MRI, you see that the wild-type cells that's shown in the central panel degrade matrigel significantly by 48 hours, but the cells with COX-2 knockdown, you see much less degradation of the ECM. And again, you can quantify this using invasion index, which is characterizing the number of cells with diffusion imaging in the matrigel layer. And also, you can do molecular characterization to show that many of the enzymes responsible for ECM degradation are downregulated when COX-2 is downregulated. So now if you have such a system, you can also do spectroscopy. And so here, what we found is that with spectroscopy, you see a significant decrease of this signal from total choline, which is something that is consistently elevated in aggressive cancers. And that is because phosphocholine, which is a membrane precursor, gets downregulated in these cells. So, of course, because these cells don't grow in vivo, we were not able to do this in vivo. But then we identified a clone which has decreased COX-2, but is not, COX-2 is not completely wiped out. And with those cells, we could derive tumors in vivo. And here you can see that these cells are called clone 13, and they actually grow in vivo, though at a much reduced rate compared to the wild type 231s or clone 2, which has COX-2 completely wiped out. So you see a little bit of COX-2 in these clone 13 cells. Excuse me, pointer's not working. So now, because these tumors, these cells can now grow in vivo, you can use imaging again to characterize the phenotypic aspects of these tumors. So as Marcus pointed out, you can look at vascularization, you can look at metabolism. And so here we've looked at two parameters. One is macromolecular transport using albumin gadolinium DTPA in the tumor. And then we've also used optical imaging to look at collagen fibers in tumors with low COX-2. And what we find is that, again, downregulating COX-2, so this is just one single enzyme, has this profound effect on macromolecular transport. So it changes the ECM. And here you can see that in tumors with high COX-2, thank you. So the macromolecular transport actually changes dramatically. It is much more when COX-2 is high, and then it reduces with low COX-2. And if you look at collagen fibers with second harmonic generation microscopy of optical imaging, we see that decreasing COX-2 significantly reduces collagen fibers in these tumors. And again, if you look at vascular volume and permeability, you see that when COX-2 is low, you see a significant reduction of permeability in these tumors. So again, this highlights how although tumors are so heterogeneous, one enzyme can have so many different effects on a tumor. So, and then of course the group from Patricia Keeley have also shown that treating tumors with a COX-2 inhibitor decreases collagen content in these tumors. And so, additionally, what you can use imaging for is also looking at single cells. And this is a concept of mechanical molecular imaging, where you can look at the mechanical properties of tumor cells, again, if following interventions. And so here, it's possible to look at the cell traction forces using polyacrylamide gel containing fluorescent beads. So when these cells exert forces, the beads get displaced, and then you can quantify the deformation and come up with changes in the traction with specific interventions. And so here, for instance, if you look at cells with COX-2 decreased or wild-type cells, you can see that downregulating COX-2 makes these cells exert less traction force. So it's as if they are less able to muscle through the extracellular matrix, again, to invade and metastasize. So this is a slide that I have up here, then just to show that, so earlier we saw that tumors are so heterogeneous, but here now one enzyme controls so many different things. So for instance, it significantly reduces metastasis, it reduces the ability to degrade matrigel, it alters metabolism, it changes permeability, it changes collagen fibers, and changes macromolecular transport. But not only that, it also shows that if you downregulate this one enzyme, you can have a profound effect on many of the aggressive characteristics of cancer cells. And of course, this is an enzyme that plays a key role in inflammation and cancer. So now, can you target this enzyme? And of course, as you know, targeting inflammation has been something that has existed since the times of ancient Greece and Hippocrates. And of course, Hippocrates showed that if you take extract from willow bark, you can reduce inflammation. But it was only until the 17th century that this was identified as a compound called salicylin. And then Bayer, of course, introduced aspirin, the wonder drug, in 1999. And then John Vane showed that this acted through the cyclooxygenase pathway and discovered the mechanism of the action of aspirin in the 70s. So, but the challenge is that many of these cyclooxygenase inhibitors, of course, inhibit both cyclooxygenase 1 and 2, so they're non-specific. And then the COX-2 inhibitors, of course, have so many other mechanisms, and the downregulation of COX-2 is nowhere near as effective as, say, using small interfering RNA. So you certainly cannot wipe out COX-2, and whatever residual COX-2 is left still can continue to confer these aggressive characteristics in cancer. So we have tried to develop ways to deliver small interfering RNA using image guidance to tumors non-invasively in vivo. And this is such a challenging target. So I'll just highlight some of the problems that can occur. So normally, it's fairly straightforward to deliver a liposome to cells that contain small interfering RNA, and your message of interest gets downregulated. With COX-2, when we did that, actually we found that COX-2 gets upregulated depending on the composition of the liposomes because these were decorated with gadolinium or iron oxide. So we tried many different formulations and finally showed that with iron oxide labeled liposomes, in fact, we could achieve downregulation of COX-2 in the cells. But for instance, if you had gadolinium on there, the COX-2, in fact, increased. And so this is a very challenging target. And for in vivo delivery, of course, again, you can decorate your liposomes with optical agents or SPECT imaging agents. But again, what we find is that a lot of these get arrested in the lungs and in the kidney and so on, and some get delivered to the tumor. And so my challenge to all you bright minds in the audience is that if you can find a way to downregulate effectively cyclooxygenase 2 in tumors, and of course visualize for personalized medicine those tumors that have high COX-2 so that they can be selected for these treatments, I think the impact in cancer treatment will be significant. So I'd like to stop there and just say that inflammation plays a big role in cancer. Although cancer is so heterogeneous and so complex, there are some targets that, if they are knocked down, have a huge effect in the aggressive characteristics of tumors. But the key is to then be able to select those tumors that are susceptible to downregulation of inflammation, and then to very effectively downregulate inflammation in those tumors. Thank you, and thank you for your attention.